didn't already know what a human was and somebody showed you a human brain and said, in this volume of substrate, how many cells can fit there? We actually really have, you would have no idea what the density of cells per unit material is, because what you can do is, and I used to do this as a grad student in duck embryos, you can take a little needle and you can make some scratches in this blastoderm like this. And when you do that, for the next 4 or 5 hours before these things heal back up, each of these little islands is not going to be able to feel the rest. And it will decide that it is the embryo and everything outside is external world. And they will make individual embryos. And you get this, you get conjoined twins. And you can get any number of them. And so the question of how many embryos are here is actually not clear. It's not set by the genetics. It's the outcome of a dynamic process of autopoiesis or self-construction. Every cell is some other cell's neighbor. They have to figure out who is part of, is this particular cell part of me as the embryo? Is it part of the outside world? And that this kind of generative medium, this excitable medium, can give rise to 0, 1, 2, up to probably a half a dozen or so different selves. And the same thing is true in the brain, actually. And I think this goes back to that point about Turing. Is we typically feel like unified individuals, but we know from studies of split brain patients and dissociative identity disorders that there's actually not clearly just one self inside the medium of our nervous system. And organs have to decide this too. Instead of making one giant eye, this one's decided to make 3 slightly smaller ones. Why? We have to understand how these different collectives decide who is in and who's out.